You are listening to Professor Megan Workman. Welcome to this lesson about nucleic acids, which are the molecules inside your cells that store genetic information. There are two types of nucleic acid molecules, DNA and RNA. In this lesson, we will describe the structures of DNA and RNA, comparing and contrasting the two. We will define what a gene is. We will explain how and why DNA replicates. That means it makes a copy of itself. And then we will describe the 4 phases of a cell division process called mitosis. Nucleic acids are built from building blocks or monomers called nucleotides. There are 5 types of nucleotides total, and they come in 2 different sizes. So here you see the purines, which have 2 rings in their structure. Contrast that with the pyrimidines, which are a single ringed structure. In DNA, there are pairing rules that keep the DNA a constant width. Notice there's 1 ring there, a 2nd ring there. So a purine always pairs opposite to a pyrimidine, and you get a DNA that is a constant width of 3 rings wide. Here's what this looks like in a chemical model. Notice there's a purine paired across from a pyrimidine, making the DNA a 3-ring wide structure. And you'll notice here that A's always pair opposite to T's and C's always pair opposite to G's. If you would like more information about the DNA base pairing rules, I refer you to the assigned reading for this lesson. Let's describe some of the other structural components of DNA that you see in this chemical model. Because DNA is a double-stranded molecule, there is specific terminology used to describe the way these backbones, these separate strands of DNA connect together. One such word used to describe the orientation of the atoms in the DNA molecule is antiparallel. And this refers to the ends of the backbones of the DNA. So here I've circled in red a phosphate group. This is at the top end of the left side strand of this chemical model of the DNA. We call this phosphate group the 5' end. But notice if we follow this across, there's no phosphate group across from it. Instead, there's an OH group. We describe the OH group as the 3' end of that strand of DNA. So 5' always occurs opposite to 3' the way an A always occurs opposite to a T, or a C always occurs opposite to a G. So now look where the other phosphate group is. It's as if on the opposite backbone it's been flipped over. So there's the phosphate group kitty corner to the other. So the 5' ends of each of the strands of this 2-stranded DNA molecule, they're occurring opposite to each other. And the arrows here, the gray arrows, and here I'm highlighting them in red, notice it goes 5' to 3', top to bottom on this side of the DNA, but on the opposite strand, it's going in the opposite direction. It's this opposite 5' to 3' orientation that is described with the word antiparallel. So DNA, because of its double-stranded nature, is described as antiparallel because of this phosphate end and this OH end. And this becomes relevant in our conversation about how the DNA machinery, how the cellular machinery replicates DNA, which we'll talk about in a future slide. All right, so here's another way to look at the DNA molecule. This is a more simplified model in which you can see the clear labels, the pyrimidines always pairing across from the purines in the middle. making the steps of the ladder. And then in light blue, we have the backbones of the DNA, and notice they're running anti-parallel to each other, 5' to 3'. But this ladder doesn't occur flat in three-dimensional space. Instead, it is twisted. So the key term to remember about the shape of a DNA molecule is that it's called a double helix. Double referring to the two backbones that are bonded in the middle, and it's twisted into this helical shape. But what about RNA? So DNA is this twisted ladder, this double helix shape with anti-parallel backbones, but RNA is single-stranded. So it occurs in a line in three-dimensional space. So here you can see the single-stranded RNA, and notice that instead of thymines in red, the T's of the DNA, the T's are missing in an RNA, and instead they're replaced with uracil, which is a different type of nitrogenous base found in RNA. Here's another way to contrast the DNA molecule and the RNA molecule. They have different functions in your cells, which we'll talk about at length in this class. But for now, just notice that their structures are different. T's don't exist in RNA, just as U's don't exist in DNA. Notice that the DNA is double-stranded and the RNA is single-stranded. Here is a table enumerating several differences between DNA and RNA that you should remember for the test. You should remember that their backbones are made up of different types of sugars, deoxyribose versus ribose. You should know that their nucleotide bases differ. DNA contains thymines, while RNA contains uracils instead. And DNA, again, is double-stranded, taking the form of a double helix in three-dimensional space, while RNA is single-stranded, and therefore you could call it linear in three-dimensional space. Now, I mentioned on the very first introduction slide that nucleic acids are the genetic storage molecules of the cell. Each byte of genetic information is called a gene. So a gene is a sequence of A's, T's, C's, and G's, if we're talking about DNA, that encode one specific polypeptide. A polypeptide is a simple type of protein, but sometimes proteins are made up of multiple polypeptides put together. So I'm going to distinguish a protein, which is a bigger structure, from its constituent polypeptides. A gene, if we're talking about DNA, exists on only one strand. So it would be the sequence of A's, T's, C's, and G's put together like genetic words. Genes are variable letters long, but it is the order of those letters that transmits the information to create all the polypeptides that make up an individual. Can RNA have genes too? Yes, of course. So RNAs also contain genes, but in this case it would be the sequence of A's, U's, C's, and G's that transmit the information. Just like the order of letters in a word tell you the meaning of the word, the order of the nitrogenous bases in the nucleic acid molecule transmits information about the polypeptide that should be encoded by that gene. Okay, so that was an introduction to nucleic acids. Now, how do these nucleic acids transmit information from one cell into another? So when and a conceptus grows up into an adult human, how does that information from that original cell get transmitted into all of the cells of your body? That's going to be answered by the next part of this lesson. So, long story short, the DNA molecule from your original cell gets copied a bunch of times, and the copies, which are faithful copies, they're proofread so that they're identical, they get put into all of the cells of your body through a process called mitosis. And mitosis here on the bottom of this diagram of the cell life cycle is just a part of the cell's life cycle where the cell is reproducing itself. The entire rest of the time, the cell is living its life, doing cell things. And then eventually it might make a copy of its DNA in preparation to reproduce. So let's review how that works. In order to make a copy of itself, the cell has to first replicate its DNA. Here is a DNA molecule that is undergoing replication. So you'll notice on the left-hand side of the screen, we have this double helical molecule where the two backbones are chemically bonded together in the middle. So an enzyme has to come in and unzip the two strands of the DNA. So it's separating the latter steps. And you'll notice the 3' ends and the 5' ends of each DNA backbone. The lighter blue is the parent molecule that got unzipped. You can see that. And then new DNA is laid down opposite. So we get a team of cell machines called enzymes that work together to build new strands of DNA, here in the darker blue color. They get built across from those original strands. And it makes a faithful copy because wherever there's an A, the cell machinery that replicates DNA knows across from A, what should it lay down? Always, always, always a T. So these DNA base pairing rules allow replication to make a faithful copy of the original. All right, so notice what's going on here. We have across from the 3' end, there's a new 5' end, and there's this continuous strand of new DNA being built into the replication fork. That's what we call that, replication fork, where the DNA is unzipping. And then out of the replication fork, we have these discontinuous segments called Okazaki fragments that get built a little at a time, and then later they'll be fused. So let's look at this in more detail. Here we have our replication fork. Here we have our leading strand. That's the continuous new strand being built into the replication fork, and these discontinuous Okazaki fragments together form the lagging strand, which is being built in fragments coming out of the replication fork. And because it's fun to talk about who discovered how this works, I wanted to give a shout out to the married couple here. They're pictured in 1968. They worked together to figure out how the lagging strand of DNA was replicated. So their last name is Okazaki, and these discontinuous fragments are named after them, Okazaki fragments. Kind of a fun human interest story behind the discovery of DNA replication. All right, so here the DNA molecule has been flipped over, and now our replication fork is going toward the right. Helicase is the name of the enzyme that's unzipping the DNA. And then we have a primase molecule laying down these RNA primers in green. And notice they have a 5' end and a 3' end. Now, this is important. Remember the 3' end of the DNA has an OH group? That's important because the next enzyme in our story is called DNA polymerase. It's the purple blob here. And DNA polymerase is kind of stupid. It can only lay down new nucleotides onto that OH group at the 3' end. So while these new, I'll call them daughter strands of DNA are getting built across from the parent strand, the DNA polymerase is smart in that it can read whether it's an A, T, C, or G and know which nucleotide to pair against it, but it can only add to the 3' end. That's why the primers were necessary. So these RNA primers got laid down and they have a 3' end to which DNA polymerase binds, and then it can start adding DNA bases. So the new strand gets elongated in the 5' to 3' direction. DNA is built from 5' to 3'. That's important. DNA polymerase adds nucleotides to the 3' end of the new strands. And then other enzymes like DNA ligase come in to replace those RNA primers with DNA nucleotide bases and binds the lagging strand fragments together. And what you have at the end is 2 copies of the original DNA molecule. And these copies are identical to each other. They're identical to the original if everything went as planned. Here's another way to look at this because it's so complex. I just wanted to show another example. Here in purple we have a helicase enzyme unzipping the DNA. That's the replication fork. It's going to travel down the entire length of your DNA molecule till it just falls off the end. And then in red we have the DNA polymerases adding nucleotides to the 3' ends of the new strands being built. You can see the black arrows showing the direction that these enzymes are traveling. Here you can also see the individual red nucleotides that are going to be pulled out of the cell and put down by the DNA polymerase bound across from those light blue parent strands of DNA. But this is a simplified cartoon, so what's missing here is the RNA primers. Okay, why would a cell bother to replicate its DNA? It does so in preparation for cell division. And there's 2 types of cell division, and we'll talk about them on separate days of lecture for this class. The 2 types are called mitosis, which happens in most of the cells in your body. It just makes a copy of the original cell to replace an old one that's aging or maybe it was damaged. This would be like in your skin cells. They're constantly sloughing off, if sloughing is a verb. You're constantly losing your hair cells, or when your hair or your fingernails grow, they're doing so by the process of mitosis. That contrasts the other type of cell division, which is useful for only one thing, making an offspring. So meiosis is the type of cell division that produces eggs or sperm. This happens in sex cells, which are the cells in the ovary or the testes that produce the egg or the sperm. So that's a very different end game in which you're going to try to half your genetic information so you can pair it up with a mate and produce an offspring that has the correct number of chromosomes. That's a little more complex. We're going to save that for another discussion. Today we're going to focus on mitosis only, which happens in your somatic cells, which are every cell in your body that is not a sex cell. So these cells just want to replace themselves with an identical copy. Here's how that works. You have the mother cell, your original cell, and here I've simplified it with a blue chromosome and a green chromosome. There's 2 chromosomes in this mother cell. Human somatic cells have 46 chromosomes, but that would be a lot to keep track of in a diagram, so we're going to simplify it here. But the process is the same, no matter how many chromosomes you're looking at. So first, before mitosis can begin, the DNA has to replicate. So it does it by that replication process we saw in previous slides. Notice in the second cell over, before the cell division begins, you have now X's, where you had one line of chromosomes. Now, when it's replicated, you have two chromosomes, and we draw them like X's just so we know that there's two of them now. And then some cell division stuff happens, and at the end you have two daughter cells that are identical to each other, and also they're identical to the mother cell. So this is perfect if you want your skin cell to replace itself. You take the original cell, which is aging, and you produce two new fresh daughter cells in its place. Next, we're going to talk about different phases of mitosis that occur during the cell division middle part of this diagram. And to remember the different phases, I've shown this squishy puppy who's learning to go potty in the proper place, but until he does so, he might have to rely on a PMAT. PMAT is the acronym for prophase, metaphase, anaphase, telophase, the four phases of mitosis. So hopefully that puppy allows you to remember PMAT. Prophase, metaphase, anaphase, telophase. Prophase is the first part of mitosis. This begins only after the DNA has already been replicated. Then that replicated DNA hangs out until something signals the cell to begin mitosis. So prophase is the first phase of mitosis. During prophase, the DNA molecules supercoil. And this means they curl up on themselves as if you took a piece of yarn and you twisted it and twisted it and twisted it and twisted it and twisted it. Or if you've ever done this with a jump rope, you twist it so many times and it kind of coils up on itself. The same thing is happening with your DNA. And when it is supercoiled like this, the DNA is garbled up together in such a way that it becomes thick enough to see under a light microscope. This is the only time that our microscopes in our laboratory here at the college can see DNA. So in lab, we will look at some cells that are undergoing mitosis so that we can see what the supercoiled DNA molecules look like. During prophase, the nuclear membrane starts to dissolve, and the cell organelle called centrioles begin building spindle fibers. So these are the centrioles, and their function is to build spindle fibers, which are proteins that are going to grab the DNA molecules, the chromosomes. So the spindle fibers attach to the centromere of each DNA molecule, each chromosome, and they begin a tug of war. Notice that the centrioles are located opposite ends or poles of the cell, and I like to think of them like fishermen reeling in the chromosome. So there's two centrioles at opposite ends, and they're both reeling in the chromosome. During metaphase, that tug of war caused all of the chromosomes to align at the equator, that midline of the cell. And as soon as they detach, because the tug of war created so much tension that those, we call them sister chromatids, detach from one another, then mitosis has progressed to the anaphase. So during anaphase, the chromosomes are traveling toward opposite poles of the cell. And also that cell membrane is elongating, it's growing, so the cell becomes oblong. Eventually, here, the sister chromatids are being pulled to opposite ends of the cell. That's what these arrows are indicating. Eventually, the chromosomes arrive at their final destination at opposite poles of the cell, and a new nuclear membrane begins to form around each set of chromosomes. The spindle fibers can dissolve because their function was to pull those chromosomes toward opposite ends of the cell. So once the chromosomes reach that destination, the spindle fibers can dissolve. And that cell membrane, which had been growing and elongating, began to pinch in on itself. So all of these things are happening during telophase, and telophase ends with the creation of two new daughter molecules. And the instant when that cell membrane pinches in on itself completely is called cytokinesis. Cytokinesis marks the end of mitosis, in which you have two identical daughter cells that are exact replicas of the parent. And they're exact replicas of each other, which is convenient if you just want to grow a new skin cell or grow your hair longer. All right, so in this whirlwind discussion of nucleic acids, DNA replication, and mitosis, you learn to compare and contrast DNA and RNA. You learned that DNA is double-stranded while RNA is single-stranded. You learned that the DNA monomers include A's, T's, C's, and G's, while in RNA there are U's, uracil, instead of T, thymine. You learned to define gene as a genetic sequence that encodes one polypeptide. Genes can be contained on DNA and also RNA. You learned about multiple enzymes that work together to replicate the DNA molecule in preparation for cell division. And you learned that DNA replication occurs during the S phase of the cell life cycle. That's separate from mitosis, when the cell is going to reproduce or divide. And then you learned the acronym PMAT to describe the phases of mitosis: prophase, metaphase, anaphase, telophase. You learned about characteristic things that are going on in each phase of mitosis. And you know that at the end of mitosis, two genetically identical daughter cells are produced.